Good afternoon, good morning, everybody. Welcome to this webinar on Vaccine Vial Monitor, or VVM. Today we have the huge privilege of receiving three huge key figures in the existence of VVM, Umit Kartoglu, Debbie Kristensen, and Ted Prusik. Umit is a medical doctor who worked for many years for UNICEF and WHO. He recently wrote the Book of VVM, Yesterday, Today, and Tomorrow, and we highly recommend you read it if you haven't yet. He's today the co-founder and CEO of Extensio and Progressio. Debbie Kristensen is a director of vaccine technology strategy and for policy at PATH. She's led PATH's work to advance VVMs for more than 30 years in collaboration with WHO. And Ted Prusik is Senior Vice President and one of the founders of Temptime Corporation. He has 35 US patents issued and 12 pending applications. He's one of the inventors of the heat marker VVM chemistry. I hope, Umit, I hope you're ready because you have the floor.
Yeah, I'm ready. You hear me well?
Very well, thank you. Go ahead.
Okay, thank you, everybody. Welcome. So if you have not checked the book, so here's the link. You can download the book in e-publication version or in PDF format that you can print. Well, VVM is pretty simple. You know, the whole philosophy is if the active square is lighter than the outer circle, vaccine is good, you can use it. When it matches the outer circle color, it's no good anymore, it's the discard point, and if you keep it more, and then the square becomes much darker. But this simple philosophy has a lot of implications on many vaccine management related policies, because if you think of all temperature monitoring devices, VVM is the only one that profoundly changed vaccine management practices and shaped and continue shaping the future of the cold chain as well. There are some approaches, some policies that in vaccine management that have only been made possible with the help of VVM. And there are some other approaches and practices that have been made more effective with the help of VVM. So without getting into too much details, I'm going to summarize which policies and which approaches that VVM makes things more effective. So the very first one is WHO policy on multi-dose vial policy, which the most recent revision was in 2014. If you think of the previous versions, you may see, you're seeing in the screen that there are 4 rules how to apply multi-dose vial policy, that VVM is a bit, not too much on the radar in the multi-dose vial policy. Actually, this is not true, because VVM gets into the multi-dose vial policy with a very critical role, which is the visual cue. Before, it was like, all liquid vaccines can be used in consecutive immunization sessions, not to exceed 28 days. Now that rule has changed. If the VVM, as you see on the left side of the screen, on the TD vaccine, if the VVM is on the label with the new visual cue and multi-dose vial policy, you can keep it up to 28 days for consecutive vaccination sessions. If the VVM is placed on the neck of the ampoule or on top of the flip-up cap, and then you have to discard these vials after your opening within 6 hours, or the way that it is described in the insert, that the manufacturers may request earlier discards. For example, this one, as you see here, which is the Cervarix from GSK, is a liquid vaccine. But VVM is on the cap, and then it gives a kind of visual cue alert that if opened, this vaccine cannot be kept, despite it is liquid, for consecutive immunization sessions. One other thing that VVM, I like to call VVM the smart expiry date. Even we have vaccine, expiry date, like here, for example, this is July 2019. Of course, it's an old photo. Before, without VVM, we all use earlier expiry first out. So if we have two vials, for example, and one vial has an expiry in 6 months and the other has an expiry in 12 months, you use the one that is going to be expire in 6 months. But if you have VVM on it, and assume that the VVM on the vial with expiry in 12 months is much darker than the one that would expire in 6 months, here VVM overrules the earlier expiry first out, and you must use the vaccine with the darker VVM, despite its expiry period is much after the other lighter VVMs contained in vials. So this is also, one extra role that VVM has overruling the earliest expiry first out principle. One other thing is the rotating stocks with the help of VVM. Even we know VVM is a kind of changes a continuous process, but in order to record, we really need to use this staging, like stage 1, stage 2, stage 3. So in a stock system, you must use the vaccines that has the second stage prior to the ones that has the first stage. For example, this photo is from Albania, and they also have a electronic system, and electronic system is arranged in a way that when VVM is marked as stage 2, it goes on top of the line. So you must dispatch stage 2 VVM first before you are dispatching the stage 1. You cannot simply select with VVM 1 in such electronic system in Albania, for example. When it comes to dispatching vaccines, so we have several aspects that we have to take into account. For example, whether there is any VVM availability, whether there is any VVM, how many batches we have, what is the VVM status and the expiry, what are the supply periods, and how many doses we need to send. It is not always the darker VVM you dispatch first. For example, here, these flowcharts are in the book. If you download the book, you will be able to, follow the whole logic. Let's go for an option that you have to dispatch a lighter VVM, even you have darker VVMs. Is there a VVM? Yes. Is there more than one batch? Yes. Are they all in the same color? No. So we have lighter VVMs and we have darker VVMs. So if the expiry period shorter than the next dispatch date for a lighter VVM, there will be a risk that the batch to be dispatched in the next shipment, short dated to be consumed on time, there will be a risk. So if you have enough quantity, you should dispatch the lighter ones. Otherwise, if you dispatch the darker ones, these ones, because the expiry period is shorter than the next dispatch date, will be expired anyway. So if you really take into account the combination of VVM availability, number of batches, VVM status and expiry, supply periods and quantity, this flowchart will really help you which vials that you really need to dispatch. Okay, one other thing is the arranging vaccines, for example, in front opening refrigerators with the help of VVM. Here is I'm so sorry for this, scrambled letters because it's coming from a different application. But anyway, so if we look at this bigger one, you put vaccine in front and the diluent behind it. If there are open vials, you put open vials front and unopened vials behind. This applies only to OPV. If you have VVM, for example, VVM, if you have a vial that has no VVM, those should be in front, and with VVMs, they will be behind. If there is VVM, you put the darker ones in front and the lighter ones behind. And when it comes to expiry, you put the shorter ones front and longer ones to the behind. And the priority goes from left to right. You give priority to the open vials, and whether there is a VVM, if there is VVM, lighter ones, darker ones, and then you come to the expiry as the last one. It's, for example, this one is from Oman, so they separated this for OPV. The ones that have the darker VVMs to be used first. For photography reasons, we took it out from the fridge, this one. And when there is an alarm, for example, high alarm, here's another flow chart, for example. Here, VVM again gets into the picture as the visual cue. For example, if there's a high alarm in a 30-day electronic refrigerator temperature logger, and if there is no VVM, you can't take the risk and you shouldn't use this vaccine and you should remove it from the fridge. And if VVM is on the label, unopened or open, it really doesn't matter. You have to check whether VVM square is lighter, so you use first. You also apply early expiry first out. And if it is matching or darker, you don't use and you remove this from the fridge. VVM also helps removing the ice from in-country transport. That is the use of cool water packs that now all vaccine carriers and cold boxes come with the life of the cold water packs. This, again, was facilitated with the help of VVM. Here's the link, how this policy was formulated based on this study that was conducted in 5 different countries. And one of the most critical policies is the control temperature chain and VVM. And I'm giving the microphone to Debbie now.
Hi, everyone. So my main purpose is to tell you about a new type of VVM, but in order to do so, first I'm going to provide some context on CTC. So as you might know, there's a long history of off-label or out-of-cold chain use of some vaccines. For example, outreach with oral polio vaccine in difficult situations, or hepatitis B vaccine outreach in Uniject. But WHO has more recently been focusing on on-label use of vaccines at ambient temperature, and they've coined this term, controlled temperature chain, or CTC, to define this use. So specifically, CTC is the use of vaccines at ambient temperatures not exceeding 40 degrees C for a minimum of 3 days. And it's just prior to administration. And this is because the main purpose of CTC is meant to be for outreach and use at the end of the cold chain. And note that 3 days is a minimum, and the CTC duration will be determined by the stability of the individual vaccine, and hopefully it will be maximized, right, as far beyond 3 days as possible. But because CTC is on-label use, the storage recommendation becomes part of the product label, and so CTC-qualified vaccines are approved by their national regulatory authority in the country where the vaccine is produced, and also by WHO as part of the pre-qualification process. And as you can imagine, there isn't much benefit to having one vaccine that's a routine vaccine that's CTC qualified when all the other vaccines that it is transported and stored with are not. One vaccine in the cold chain and the others not in the cold chain doesn't make sense. So the focus for CTC is therefore on vaccines used in campaigns and special strategies like outbreak response or outreach to schools or homes. And lastly, I just want to mention there's a WHO CTC working group that to date has been reporting to WHO's Immunization Practices Advisory Committee, and this is the strategic roadmap you see up to 2020, and we're now developing the updated roadmap for 2021, and Analia Kahn's the point person for this work, and I believe she's on the call. So currently we have 3 vaccines that are qualified for CTC use. We have Serum Institute's Menafravac, Gardasil HPV vaccine, and Shankol oral cholera vaccine. A number of other vaccine manufacturers are in the process of qualifying their existing and their pipeline vaccines for CTC use as well. And I'd like to make TechNet members aware that not all vaccines are heat stable enough to qualify for CTC use, even if formulation improvements are made to optimize their stability. So we're always going to need the cold chain, but we should really maximize use of CTC qualified vaccines to meet countries' coverage and equity goals and to ease logistics. So now we'll get back to the VVM. So the current version of the VVM, it's a marvelous tool and it changes color in response to cumulative heat exposure, but it doesn't respond rapidly enough at higher temperatures for some vaccines, especially the CTC ones, the more heat stable ones. And the problem is that vaccines can deteriorate really rapidly at these very high temperatures. For example, the proteins might denature. And that's why when using vaccines in a CTC, we use a threshold indicator, or a TI. And these TIs react rapidly above a defined threshold temperature, and they provide that extra assurance that's needed when vaccines are purposely kept at ambient temperatures. So currently, when we're using vaccines in a CTC, we're using standalone TIs to monitor the vaccines, and they're being placed in the carriers, as you see in the photo, or in the cold boxes. And so the indicator is on the card that you see on the right now on the top, and it turns black after just a few minutes when the temperature has exceeded 40 degrees C. So the use of these cards is helpful, but it's also been a barrier to CTC use of vaccines because the cards need to be procured, they need to be stored below the threshold temperature, and distributed. They can be reused as well, so reverse distribution is also advisable. But in addition, they're a major component of the training needed for CTC use of vaccines, since now those managing the vaccines need to read both the VVM and the TIs. So here we go. The next generation VVM offers a good solution, and you can see it here. It's integrating the TI with the VVM, as you see in the photo on the right. The indicator reacts just like a VVM up till the threshold temperature, and then if it's exposed to that threshold temperature, it immediately converts to endpoint. It goes darker beyond the color of the reference ring. And WHO has already developed pre-qualification specs for this indicator. And so I'm now going to turn the presentation over to Ted, who will tell you about his company's efforts to develop a VVM TI.
Hi, Debbie. Thanks. Hello, everybody. Thanks for joining the webinar. So we call the combined VVM and threshold indicator VVM plus because it is the VVM plus the threshold indicator. The picture you see here is the first pre-qualified VVM plus, the heat marker VVM plus 250. And so what's the value of having the combined indicator? Just as Debbie said, sometimes the logistics of getting the threshold indicator out to the field, to the edge, has caused some issues. This slide shows a comparison of a regular VVM on the bottom as a function of time and temperature. As you know, the VVM gradually and slowly and irreversibly changes color. But one of the criticisms of VVM from the beginning was that at high temperature it doesn't react fast enough. And so the addition of the threshold component, it was placed at 40 degrees C to be compliant with the current CTC initiatives. Once you hit the 40 degrees C mark, over a few minutes, as Debbie said, the indicator rapidly and irreversibly darkens. So that it's clearly that the temperature has gone above the threshold point. So the VVM reaches its end, VVM plus reaches its endpoint either if it's been kept for a long time at some temperatures and works as a standard VVM, or if it's gone above that threshold temperature for a short period of time. The next slide, please. There you go. So in order to pre-qualify the VVM plus, as we call it, specification had to be drafted for the combined VVM and threshold indicator, which was done. And we've pre-qualified the VVM plus 250. And we've also pre-qualified a VVM 250. The VVM 250 is a lot more stable than the VVM 30, about 8 times more stable. And we also pre-qualified the threshold indicator as part of the PQS pre-qualification. We're about to submit VVM plus 30 to the PQS laboratory to get it pre-qualified also. Next slide. So the VVM plus is shortlisted on the Vaccine Innovation Prioritization Strategy called VIPS. There were 24 innovations to begin with. Under the phase 1, 9 of the innovations have been shortlisted, so the list is shorter. And by the end of March, 3 or 4 of the innovations will be the finalists in the system. The VIPS program is a coordinated program between UNICEF, WHO, Gavi, Gates Foundation, and PATH. So we're really proud that the combined VVM threshold indicator is one of the so far shortlisted items. Even if it doesn't make it through to the finalists, it doesn't matter to Temptime. We're still going to commercialize and have the VVM Plus available. Next slide. So we're switching gears a little bit to the future. The future has barcodes in the distribution of vaccines as well as other commodities and drugs. What you see there is a 2D data matrix barcode. It's a GS1 compliant barcode. GS1 is a global standards organization, and 2D GS1 data matrix barcodes are being adopted by many countries as their standard barcode versus a 1D barcode that doesn't contain a lot of data. 2D barcodes has high data density and a high security level. So the 2D GS1 data matrix is pretty much the standard, but not in every country. It is being adopted and recommended by many global organizations, including UNICEF, WHO, et cetera. Next slide. As a show of this, Gavi announced in, I guess it was October, that GS1 barcodes are going to be required on secondary packaging of vaccines. All tenders after October 1st will have this as a requirement, and it must be implemented by 31st of December 2021. So why are people going to barcodes on secondary packaging as well as other packaging levels? It's to help with track and trace systems in different countries and across countries and globally. Track and trace is a way to monitor and track a commodity from the time it's manufactured at a manufacturer all the way through the supply chain and give you an electronic means to capture that information and use it for your information systems. WHO is also developing a track and trace policy brief. I'm not sure when it's due to be presented, but I believe it's very briefly. So again, barcodes are in the future. It's a requirement coming up now with Gavi on secondary packaging for vaccines. And next slide, please. In the United States, 2D barcodes have been required on vial-level vaccines, vials and pre-filled syringes, for the last years. It's more than 80% compliant with the 2D barcodes on vials and pre-filled syringes. It's up much higher than that now. And if you look on the left-hand slide there, part of the slide, it says, these bad boys are a game changer, scaling 2D vaccine barcode scanning. This is from the United States CDC back in August of 2018. The amount of information that you gather by scanning barcodes on vials, pre-filled syringes, is huge. I mean, it's a lot of information that's not available now with all the track and trace regulations and certainly within your countries. The next slide. This just shows that the 4 prime suppliers to the U.S. market have put 2D barcodes on not only the pre-filled syringes, but on vials of vaccine. So again, the high level of compliance in the United States based on a requirement that this be done, and the requirement was put in place through the AAP, the American Association of Pediatricians. They felt the need for this, and it's been implemented in the U.S. and in some other countries as well. Most notably, Turkey has 2D barcodes on just about every drug it has in the country. Next slide. So we're taking the 2D barcode approach one step further by using the VVM chemistry inside the 2D barcode. To us, it doesn't matter whether that barcode is on a package or on a vial or on an upper level in the system. Next slide. Next slide, please. So the idea is to use the same chemistry that's in the VVM. It's printed in a specific region inside the 2D barcode. And with a scanner or a smartphone, one can capture the 2D barcode information that's stored in that barcode, as well as get a snapshot of the color level of the VVM. With a good level of sophistication, one can get some remaining life of the barcode. We're not there yet with respect to getting that as best as we can. As many of you know, we were purchased by Zebra Technologies about a year ago, and they're helping us to tune that algorithm. So it's an enhanced value of the barcode for stock management, patient safety, and anti-counterfeiting, because the VVM is really a part of anti-counterfeiting effort also because of its unique thermochromic properties. So again, you can rapidly read it, and the data that's captured in the field can be sent to the cloud and into databases and blockchains, as you all know. The next slide. Let's see if the animation works. There we go. So as you see, it's the same chemistry that's in the VVM that's encoded in the 2D barcode. Thanks. Next slide. So this is a Zebra smartphone that has the OneScan app in it. What you'll see is we're going to read a barcode on a vial that has, in fact, a 40 degrees C threshold indicator on it. It's being put on a hot plate. Once it's put on the hot plate, the indicator is going to turn color very rapidly because it's about a 90°C hot plate. As you see, the one corner is turned color. And now it says contact supervisor. It's detected that the indicator has been expired. So again, all that information, the GTINs, batch number, expiration date, serial number, GPS location, that's all been captured by the scanner and pushed to the cloud. Next slide. So just an idea of what that looks like on vials. You can see them by eye, but obviously it's not meant to be looked at by eye like the VVM. And we believe that the early adoption of the 2D barcode with the VVM embedded will include and maintain the classic VVM in that first presentation. Next slide. And just an idea to play the video. So one can have the technology, whether it's a VVM or a threshold indicator on a high level of packaging, a smaller level of packaging, a large vial, a curved surface, a smaller vial, a pre-filled syringe, or even down to the ampule level. So the technology exists to measure, monitor measure barcode indicators down to the lowest level on the ampule, which is a huge challenge and a huge achievement. Next slide. So with all the data that's, all the VVMs that are used around the world, there's not much data that's captured on them. And when you capture the data of the VVM, there should be a way to now link that to an HMIS system. The next slide. So if one has an immunization card with either a barcode or a way to identify the particular child that's getting the vaccine, you can try to link the HMIS data, the health information data, along with the logistics management information that you're getting from scanning the barcode, and actually track what vial has gone to which child and at what time and when did they receive that vaccine. Having the VVM as part of it gives you the information about the quality of the vaccine or the temperature sensitivity of the vaccine, as well as the information that's gathered from all those data. Next slide. Thank you from all of us. Umit?
Thank you very much. Umit, is there anything you'd like to add? I'm unmuting you.
Okay, thank you very much for your patience. I know that we presented the VVM implications and its assistance in VVM in vaccine management policies very briefly, and the future of VVM, how it is shaping the digitized track and trace systems. Now, for the rest of the time that we have another 20 minutes, we would like to open the floor for comments and for questions.
There are already a few questions here.
Yeah.
Shall we start?
Yeah, please, go ahead.
Let's go. So, Geneva, as everybody knows here, why TD vaccine not qualified for CTC?
I can respond to that.
Go ahead.
So it is a priority for WHO and the working group. Absolutely. We're just working with manufacturers to try to do this. In some instances, the preclinical studies required are a barrier because it would require a lot of preclinical testing in order to receive approval. But we are trying to move that forward now, especially since it's sometimes taken on outreach with HPV vaccine, which is already pre-qualified. Thanks.
Thank you very much. I would just like to add, in case some participants would like to participate and are not aware of it, there's a chat box at the right hand, so you can type your questions there. The next one is still from Serge. In the barcode, can you check the type of VVM?
This is Ted. Yes, the barcode will have information about the VVM type, and so it has all the kinetic parameters of the VVM embedded in it. There are GS1 standards for the way the data is presented, so that it can be interoperable. It identifies the VVM type and its properties. So yes, good question, and yes, that's been taken into account on a global level.
So I took back to that slide that here, for example, it says this one is the VVM7. Did you hear?
Yeah.
Okay.
Thank you.
Okay. Let's move to the next question.
Next question. Paul Blum from the WHO. Can any scan program be used or specialized programs? His iPhone did not read it. So it seems Apple has an issue with the VVM.
I'm not sure I understand the question.
If it's the scanning program that is on the phone, it seems that the one he was using on his iPhone did not work. So is there any issue with any specific scanning programs, or is there any specific program to use? Paul, correct me if you want to reformulate your question.
So we are developing a specific app to read this. Mm-hmm. So we're only standing behind our app that we're developing. I know some people have done some app development. I'm not sure what they are, but the one that we are working with should be able to be used on a smartphone, iPhone, or Android, as well as with a scanner, as you saw in the ones that go very quickly. They are using a red light. We also have white light under development, so it should work.
Okay, so you need a specialized scan program that is developed by you.
Yeah, and that'll be one issue to address. Yep.
Okay, thank you. Another question from Sibenu Derek. Is scaling up the barcode VVM technology a possibility for developing countries in the near future?
We hope so. We're trying to organize pilots right now, and we're working on that together with WHO as well as UNICEF and other partners. So we hope to get some proof of concept in the field. We've done some small studies here in the United States with a major distributor of healthcare products. And we're actually in an FDA-approved pilot study right now with a consortium led by Remedy. It's not for distribution from manufacturer through to end user, but it's for tracking the products from a hospital, inside a hospital system, all the way down to the patient and across hospital systems to check interoperability. So, and we believe that, yes, developing countries, this should be available to them also, yes.
Thank you very much, Ted. Question from Pamela Mitula. What material is the barcode made of? Does it easily peel off when exposed to moisture?
So the materials are exactly the same as the current VVM. The VVM Plus is going to be a label, a dot label, but the VVM substrate, the label substrate is going to be the same substrate that we have now on the VVM. So whatever issues you have with moisture is still there. I mean,
okay. Thank you, Ted. Next question from Sally Embray. Will VVM Plus only be applied to CTC-approved vaccines?
So
Ted.
Go ahead.
So the example we showed you was a VVM plus 250. That vaccine is going to be labeled for room temperature storage. So from that point of view, no, it's not just CTC vaccines. Debbie?
So we are looking at that, WHO is looking at that as more CTC vaccines become pre-qualified. I think it will be a WHO decision to specify VVM plus on CTC vaccines as they specify VVMs on all vaccines now. There is a possibility that the VVM TI could be used on all vaccines, but I think the extra cost might be prohibitive to go in that direction. So it's something that should happen in the future, I would hope.
Thank you very much. Serge Geneva is asking if we don't have the barcode reader, can we check the type of VVM?
Yes, this is Ted. Every VVM has, if it's a dot, the VVM type or category is printed, is on the VVM. And if it's full label, it says it right underneath. It says heat marker VVM 7, heat marker VVM 14, etc. So every VVM category is on the VVM or with the VVM.
Okay, thank you. Snehal Singh is asking, is there any independent study to prove the relation of VVM decay rate with antigen decay rate?
This is Umit.
Mm-hmm.
Hello, do you hear me?
Yes, we can hear
you. Well, there are some early studies that shows the correlation between VVM color change and the degradation of the vaccine. This was mainly done by NIBS in UK during the early implementation times of VVM with the oral polio vaccine. There are some other studies that were done in different campaign settings, again with the oral polio vaccine in Mali, in Chad as well. You can find all these studies and their summaries in the Book of VVM in the section that is called the science behind. But there are studies that shows the correlation of vaccine degradation and the VVM color development.
This is just to pipe in also that when a vaccine is pre-qualified, stability data must be taken at 37 degrees C, and normally the typical storage temperature of 2 to 8, and many vaccine manufacturers also do 25 degrees C. And so those data that are provided to WHO as part of the pre-qualification process are used to help select the VVM type category. So there is an on-paper correlation done for each vaccine that is pre-qualified.
Okay, thank you, Ted. Thank you, Umit. From Paul Blum again, can you explain if different VVMs are used for the CTC vaccines given they have different durations of 3, 4, and 14 days? And does the VVM for Gardasil indeed, sorry if I mispronounce it, indeed turns after 3 days?
Hi, Paul, thanks. So these are very heat-stable vaccines, and they all use VVM 30, both the HPV and the oral cholera. And the VVM changes as it normally does, and then the separate threshold indicator is used to monitor whether or not the threshold temperature is breached. But the number of days in the CTC has to be monitored by those managing the vaccines. And so they have to have a system in order to log how many days the product has been in a CTC. So right now it's a separate system. The VVM doesn't integrate time like that. It's time and temperature. So a separate system has to be devised to capture time. Thanks.
Okay, thank you.
Just one extra comment there with respect to CTC and the vials. I mean, if a vial had a barcode on it with a specific serial number and it was scanned as it's put into the field, you can know what date and time that was put into the field and the serial number. So you'd have a way to track that electronically also. When the vials are brought back, you could scan it and make sure that they've come back. And if you're in the field and scanning and you have good coverage, you'd be able to calculate if you have any more time remaining within that allotted period.
Okay. Thank you, Ted. From Omileye Toyobo, this is a long question. Hi, Umit. Great presentation from Omileye Toyobo at CHAI. Please, what is your take on the use? What is your take on the use of the freeze-free vaccine carriers in terms of the unintended consequence that it could result in heat damage while trying to prevent freezing? During a study, we noticed that it takes about 3-4 hours for the temperature in the container to cool down to within the safe zone. This may leave vaccines that are already in stage 2 exposed to some level of heat damage. Should this be a concern, or are we comfortable with that small window provided we prevent the greater evil of freezing, which damages vaccine faster?
Is this in refer to freeze-free vaccine carriers? Yeah. Okay, so
yes,
if I understood correctly, because I'm not sure that I understood the question correctly.
Do you want me to repeat
about, okay, can you read it once more time, please?
Of course. So, what is your take on the use of freeze-free vaccine carriers in terms of the unintended consequence that it could result in heat damage while trying to prevent freezing?
Yeah. Well, I believe that it is pretty slim chance of having a serious high temperature exposure that would put vaccines at risk. Of course, if you are putting a vaccine that is, for example, 95% life is gone, that you have only 5% very, very close to the end point. And if it is VVM2 or VVM7 type of, containing vial, and then the risk of those ones being affected compared to VVM14 and VVM31 would be much, much higher. I do not have any data that I can refer right now, in my hands to explain what would be the, expected time for having the higher temperature excursion. However, if I take you back to the study that was, let me go back, the one that came, which this one is not the freeze-free, how to say, freeze-free vaccine carriers are called boxes. So here there was a kind of a calculation that if you repeat the test exposing the cold box with cool packs inside between 2 and 8 degrees, preferably 5 degree, which is cool, which doesn't have a latent heat like the ice has, 4 times for 48 hours at 43 degree constant temperature. So here are the images of VVMs. What would happen? Again, 43 degree constant temperature, 48 hours exposure, 4 times repeated. So as you see, even VVM2 doesn't come to its end point. So VVM2 here, for example, VVM2 means 37 degree in 2 days, it reaches its end point. So after this exposure of higher temperature, the remaining days of VVM2 will be 1.1 day, which is not even 50%. You didn't. So this VVM is still in stage 1. It didn't even reach stage 2. So we're speaking about 43 degree constant 48 hours 4 times. I don't think freeze-free cold boxes or vaccine carriers would ever have such higher temperature excursions. So I believe that it is quite a safe move, and the vaccines being impacted with the higher temperature should not be a concern at all. Debbie, would you like to add something to this?
Yes, I'd just like to add that I think freezing is usually a greater risk than heat exposure for most vaccines, and also that WHO, of course, will have or has pre-qualification protocols for freeze-free carriers, and so that includes testing and field evaluation. So I would assume that those risks would be taken care of by virtue of the spec set for the carriers and the testing that has to happen in order to pre-qualify them. Over.
Yeah. Thank you.
Thank you very much. Several participants are asking if we can share the presentation at the end of the webinar. We're going to share the presentation as well as the video. Everything is going to be shared on TechNet this week. Next question, still from Omileye Toyobo. Does water damage hamper the ability to scan the VVM Plus and the barcode in case it is slightly peeled off, which happens due to condensation in the field?
So scanning barcodes are, the barcode has to be pretty good in order to scan it. The 2D data matrix barcode allows, I think, for up to 40% damage of the cells, and they can still get an accurate reading. So it depends on the state of how bad it is. I think you could see that, when I'm sure you scan barcodes and when you're shopping and they read pretty well. Damaged barcodes are a difficulty in any industry. It's not going to be different here.
Thank you, Ted. Next question from Snehal Singh. Even after second barcoding, classic VVM will stay at Vial to manage the Vial accession. So what would be cost contribution of this barcoding?
That's a good question, and we still don't know the answer to that. WHO is, and the VVM is a main part of our business. We want to maintain the classic VVM, and we're not sure on how the addition of the VVM chemistry and the 2D barcode will impact the cost. Of course, for developing countries, we'll try to minimize that cost as much as possible. We have to look at what the value is to the country with that mode of gathering data. We're also looking at, quite honestly, what we tried some years back, reading the VVM and the barcode in line So not putting the chemistry separate into the 2D barcode, but just capturing the image on the side. When Temptime was trying to develop it ourselves, that was a big challenge. Now that we're part of Zebra Technologies, who specialize in reading barcodes, we're working with their decoding team to look at how feasible that is. In other words, can we use just the 2D barcode and the current VVM and get good data? That's good when you have real estate, enough space on the label. The idea of integrating the chemistry into the barcode itself was to save precious space on vials. So excellent question, and we're looking at both ways to read and capture VVM data, all with the idea to minimize the additional cost or any additional cost, and bring as much value as we can to the supply chain and to the immunization program.
Thank you, Ted. It's already 5 o'clock. Do we have the time for a last question?
Of course, go ahead.
Okay, so considering that in some places there is no internet connection, can we save as picture the QR code for posterior analysis, or is it not recommended?
So the program to decode will be in the instrument that you use, the device that you use. The data will be saved and will be pushed to the internet once you get it to an area that has the capability to do so. So the data is stored and then it's forwarded once you get to capability.
Great, thank you very much. So there might be maybe 1 or 2 questions left. No, I think if all the participants here are interested in asking more questions, you can use the TechNet forum. We're going to post the video, a link to the video as well as the presentation. And yes, if you have further questions, you can use this thread, and hopefully Debbie, Ted, or Umit will be able to answer your questions. Thank you very much. Thank you very much, Umit, Ted, Debbie. Do you have any? Would you like to add something before we close?
Thank you, everybody.
Thank you very much, and have a good day, afternoon, evening, wherever you are. Thanks again. Bye-bye.
Thank you. Bye-bye.
Thank you. Bye.